enough to do metabolism or forced to, one of the two. I'm sure you've heard all about it, and you'll hear more about it, and me, and what I do to students particularly. If you survive, you'll probably get a medal. Key thing about what we do is trying to help you ready yourself for your careers. Now, this unit is a level 2 and requires a fair bit of focus on your attention during the semester. Some of the things we do very deliberately. Some of the things that happen, like a 4 till 6 p.m. lecture, is out of my control. It was either that or a 6 till 8 p.m. lecture, which I'm not sure you'd be happy with, neither would I. This particular campus is rather full, and hopefully when the dust settles, you're welcome to go to any campus for lectures. It doesn't matter, even though there's a requirement to do that, and Parramatta's pretty full. They're sorting out, hopefully, a better lecture theatre for us up there. But please don't be scared to go to other lectures and other campuses. If it suits you, it suits me fine. It doesn't matter. The same material, the same dumb jokes, the same everything as we go through. Today I'm going to take you through this little animation, which is kind of critical, and I'll come back to it at the end of the semester. It's a bit too big and clunky to run in views, but it has kind of an overview, and I like the conceptual overview first, because unlike one of my third year units, Advanced Cell Biology, which is very illustrative, pictorial, lots of animations, lots of beautiful images, as some of your colleagues in here have done that unit successfully, this is a very conceptual topic area, unfortunately. And because of that, we run a number of other support programs, including PASS. Now, Michaela, just here, is one of the PASS facilitators for this campus. It's online. Some of you are involved in PASS, but I can tell you from experience, and I'll give Michaela a chance to talk to you in a minute, it's very important to make sure you keep up to date with this material, okay? You cannot cram metabolism. Oh, yes, I can. No, you can't. You might think you'd be able to go and do that for the questions in the exam, but you can't cram the concepts, which are 4 short answer questions that deal with the concepts, which means you need to try and bring those together. I'm going to take you through this video, then I'm going to take you through the material I put up on views. You'll notice I put a little red dash box and a white box in there for you to fill in. That's the last time you'll see the white box with the dash red lines. Every time after this, you'll have to look at the slide and see where you'll need to add things to. Now, it's not usually a lot of writing, but it's usually critical writing. Why is that? Well, we know from learning research how your brain learns by writing the physical process, not typing, not just listening, but writing in actually makes more neuronal connections, which we talk about in Advanced Cell Biology. That will help you learn the concepts. That's why I ask you to write a little bit. How many slides did I put up there already? 16, 17. I don't normally put up more than 20, and that's for 2 hours, roughly 8 to 10 slides per hour, which means we have time to talk, time to discuss. You'll notice it went up at 1 p.m. on Thursday. That's going to happen each week. Why is that? Because the lecture at Parramatta finishes at 12. Give a little bit of time. It'll pop up ready for you to have a look at it before the lecture. Because when we go through it in the lecture, if it's the first time you see it on the screen, it's going to look like some circuit diagram. You have no idea what's going on, do you? So have a look at it. Doesn't mean you have to read it all in detail, but just have a quick look before you come in here. I'm going to take you through the basic concepts of metabolism, why we do what we do, and everything in my units is very deliberate. We do things very deliberately to try and help you. Why is that? Well, some of you may or may not know that I spent quite a long time in CSIRO and overseas, not just in university. 17 years of CSIRO, hard labour, doing all the things we do around the world, developing new science, and as an employer, and taking students in, dealing with them, getting them ready for other employment. So I know what you need to be ready for if you're looking for a job. Because I interview many. I say, I don't want you. I don't want you from the sandstone, because you just fill in boxes. That's most of your lab manuals, isn't it? There's a box, fill it in. Not in this unit. Not in my third year unit, is it? You are writing what you need to do when you get to the real world. And as I said, everything's deliberate. Now, I'll let Michaela out of her torture and let her talk about what's going on. I'll come back to this video so that we can talk about the concepts.
So many people.
No, there's only a handful. You wait till we get 400 or 500 in there. Giving a conference in Japan many years ago, 30,000 delegates in the room looking at me. 30,000 people looking at you. There's only a handful, and they're all your colleagues.
Sure. Okay, hi guys, my name is Michaela. I'm the PASS facilitator for Metabolism this semester, specific to Campbelltown. So most of you guys are in your second year, I'm guessing, but who has heard of PASS before? Who's attended PASS? Okay, well, this semester you will. So, yeah, most of you in your second year, but just to recap, PASS stands for Peer Assisted Study Sessions. They're weekly 1-hour group study sessions facilitated by a senior student like myself. We basically review lecture and tutorial content. Very flexible, so you guys actually decide what you guys want to do each session, what you guys struggle with. We learn study skills and techniques, and I'll let you know like the study skills and techniques that I used for the unit that helped me. Sessions are free and no registration is required, so you guys can just turn up. So on average, students that attend PASS achieve higher grades, so 7.7 grade points higher compared to students that did not attend PASS. So these are just some stats. PASS also increases your confidence in tackling obstacles and questions in exams. You can also make friends, which is a good thing in every unit. You can form study groups and things like that. So just to recap, quick presentation. PASS is free, weekly. Sessions start week 2. No registration required. You guys can just turn up. If you can't turn up one week, it's okay. You can turn up the next week. But try coming each week so you're up to date with everything. Session and times are on views, but here's just a little rundown of it. So this campus, I run them on Friday 1 to 2. So I think for you guys, it's before or after your pracs. And that's the room number. And yeah, that's it. More information is on views, and Mark will probably talk a bit about it. And that's it. Any questions? No? Okay. Thank you. See you on Friday, week 2.
Thank you, Michaela. I'm sure they'll have lots of questions as we get rolling through the unit. Now, as Michaela points out, it actually helps with getting through this unit. And as she pointed out in the data, it actually helps a great deal with confidence. Now, this unit particularly is dealing with concept areas, okay? And why I want to take you through this little video is to get in your heads a visualization, because some people are audible learners, some people visual learners. Now, I'm more of a visual. I need to see things in space and time to understand how it works. Metabolism, if you look at the textbook, have you seen Leninger? I stick it on reserve in the library. Seen how thick it is? Hasn't changed. I used Leninger as an undergrad myself. I still have that original copy. And I actually have every edition since, because the publishers keep sending me copies all the time. Now, it's not because I'm going to torture you the same way I was tortured, because what we were doing is trying to memorise every pathway. And some of the pathways weren't resolved. Molecular biology was just getting started, so we didn't understand gene expression, all those other elements. Memorising it was totally pointless. Totally pointless. What I'm trying to get across to you, apart from your memory stick, to get across to you is the key regulators, the key concept. How does your cells take in the food you've just slipped in before you came into this room, or munching away right now, turn that food stuff into something that's you, either energy or muscle or bone. We talk about those things in advanced cell biology. But you're managing that food stuff. And one of the critical things to keep in mind, you as a human are good at, and I'm going to tease you all semester, turning sugar into fat. That's what you do. Carbohydrates, you turn into fat. If you don't make them turn into energy, which you're about to see in this video, you will turn them into fat. That's what you do. Fat can be used for energy, but it doesn't make things except lipid bilayers, which we're going to see in this animation as well. Now, if we can understand those concepts and work through them, and as we go through them in all my lectures, as some of you know, I like interaction, I like questions. And even though I'm down here and you're thinking, oh God, it's too scary to ask a question, there's no question that's stupid as far as I'm concerned. If you're not sure, just yell out. I don't understand that bit. I'm happy to go over it again. We have time in the lectures, but if we don't get it right in the lectures, you're going to have trouble with everything else. And the key thing, as I said, is not just about understanding the pathways, it's who's regulating what. So that when you do molecular biology, when you do cell biology, when you do anatomy, physiology, you understand what drives things. When a muscle works, how does it work? Why does it work? What forces that motion? When you're in the gym, when you're running, when you're sleeping, what are you doing? And all of those things are becoming more important, particularly because in this part of the world, obesity is on the rise. It's overtaken diabetes. 1 in 3 now are obese. Now, as a graduate from this unit, it'll be your role to try and convince the public to try and manage their disorder. Now, obesity is not something, oh, just stop eating so much and exercise. You'll see towards the end of the semester when I talk about how your brain and your stomach control what you do. Your stomach is feeding your brain information, the same kinds of information that drugs do. It's the same pathways. Drugs that cause you to change behaviour are working for the same pathways that food does via your gut. And you'll see the recognition of that and how you can potentially manage it. Let's get back to this animation. That's a mitochondria. We can see some of those in advanced cell biology. These characters move around the cell wherever the work is. They're mobile. You have them in almost every cell, except maybe your red blood cells. They kick out the nucleus and the mitochondria. They only live for a short time, about 140, 150 days. Some of your cells only live for a week or so. That means their turnover rate from stem cells, as we talk about in advanced cell biology, to a mature cell doing the work, particularly in your gut, is very, very short, which means the potential for tumours. And you'll see even energy can tell you whether a cell's becoming a tumour cell, what it's doing with it. So here's a mitochondria running around a cell, being driven to where the work is, and there's motor proteins that wander along dragging this mitochondria, which we'd see next semester. This thing, just go back a tad, that blue thing is a long chain fat, long carbon, maybe 1 or 2 double bonds in there, lots of hydrogen, an acid function on one end. That fat molecule has to go into the mitochondrial matrix to be broken down through beta oxidation, 2 carbons at a time. But while it's a fat, what does it want to do? There's my long chain fat. It wants to aggregate with other fats. You have to keep it separated. That's what those yellow things are, proteins to keep them separated, to move them around the cell. Where am I want to move it to? Well, into the mitochondria, through a particular channel. That channel process I'll take you through in detail later on. A lot of turnover, a lot of messing around to activate, deactivate the fat, get it through the outer membrane of the mitochondria, you can see there, here, via the intermembrane space into the inner mitochondrial matrix. A lot of messing around to get that fat molecule in there to turn it into energy. And that mostly happens in muscle cells. They will use a lot of fat for work when they're required to. I go into the mitochondria now, and I've got all these proteins. What are these things? These things are embedded in the lipid bilayer. That's what you see here. These little ball things are the phosphate head groups, the polar head groups forming a lipid bilayer. You can see how mobile they are in this beautiful Harvard animation. And there are proteins embedded in there, allowing transport of molecules, selectively dealing with it. We're now inside the mitochondrial matrix. That looks like DNA, doesn't it? It's a mitochondria. They have their own DNA, just like bacteria. But all these things are proteins, lots of enzymes. There's lots we're going to talk about, but some key regulators we're going to focus on. Who controls it? Who's the foreman? Who's the executive that makes the decision to get that pathway running? And I'm going to use lots of analogies in this unit, particularly things like building a car. You know, you have a production line. When everything's lined up more efficiently, like a German system, you can build it faster, more efficiently. But someone says how fast to go, how many to make. There's a feedback to say what to do. All the workers, all the robots just do the work. Who turns them on and off? Who speeds them up and down? They're the ones we're interested in. That alters your metabolic rate. That burns more fat, for example, or doesn't. You lay down more fat then. All the things you don't want to do, particularly if you're going into summer, trying to beef up and look good for it. Here's all our proteins. This one is one big one we're going to see later on. You might want to note, just now, PDH, phosphate dehydrogenase. It's going to take those carbon molecules, activate them, ready to make ATP. But there's a long way to go to make that ATP. How do we make that ATP? Well, every time you break a carbon bond, how many of you live on a farm or have a combustion stove you throw a log of wood in? 1, 2. Nobody else has a combustion stove? What happens when you burn a log of wood? Flames. But you break carbon bonds. Log of wood's just lots of sugar moieties, all hydrogen bonded. What do? You burn the carbons. You break the bond. You generate heat energy. Every time you do this in a mitochondria, you generate electrons and protons. And you're going to move those protons around to make sure you don't burn your mitochondria or you. Otherwise, you'll be a piece of burnt wood, wouldn't you? So you're trying to manage that with enzymes. Hence why you do FP and G for a start. If you survive that, you come to me. If you survive this, you'll survive anything. But to get that electron into energy, we have to go through these beautiful molecules. See those rotating characters there? We're going to generate these electrons. See those sparking things? We're going to see a whole complex, and there's actually 4 proteins that help move electrons through the inner membrane of the mitochondrial matrix. They take the electrons from the broken carbon bonds, move them through the membrane. And as they do, they help pump out protons to the inner membrane space. And that generates like a battery. There's my 2 membranes, there's all my protons on that inner membrane space. Because it's more positive there and negative in the matrix, they want to come back in. So I'm moving all these electrons, managing the protons, which is actually important for cancer as well, because this generates reactive oxygen species. There's a lipid bilayer. This character you're going to get to know, he's called cytochrome C. He carries electrons in the intermembrane space from one molecule, jumps it to another. This character, the advanced cell biology students found out, as soon as that guy is loose in your cells, it will cause cell death, called apoptosis, programmed cell death. A cell will die so that everything is recycled. It won't blow it up, but everything's recycled. And all of your cells go through this. Some faster, like your gut, some slower, like your bones. All of them go through apoptosis. Otherwise, you look like a burnt piece of wood. There's cytochrome C moving electrons to complex 4. Just transferred them. What happens? All the protons zooming around. Here's another complex protein. This is the guy that does all the work. This thing is called ATP synthase. It has a rotating functional domain, we just saw a moment ago, a bit like a water wheel. See that water wheel there? That is a protein that's actually rotating just like an electric motor. Instead of electrons running around, those little bright spots are protons coming back into the matrix, literally rotating the wheel just like a water wheel does. As it rotates, it spins this axle here. As it does, it changes the head group conformation. It grabs ADP, ATP, and literally vices them together to form ATP in the mitochondrial matrix. That's not what we need ATP. We need it outside in the cell. So we're going to have to see how that works. So there's the rotation. ATP synthase, you can actually label with a fluorescent molecule and see it rotating inside a cell. You can actually see the process of that rotation, which is all the cell biology and molecular biology we play with. There's ADP in, ATP out. Now, one of the key things here that I need you to think about as we start the semester this time of year is it's cold outside, isn't it? Been the last few days. Today it's hot, but it's not really hot, is it? It's only like 20, 23. Oh, it's hot. Why do I feel so hot? Because some of those electrons that are going out, ATP forming because protons are coming in, are being disrupted in your mitochondria, particularly in certain fat cells. So instead of generating ATP, the mitochondria in those fat cells generate heat energy. They literally warm you. So during the winter time, you have lots of these protons, I'll talk about later on, that generates heat energy in those fat cells. When you're first born, that's what you have as well. That's why you come out with no clothes on. But there are certain places you generate that heat energy. Because you're in winter, you have more proteins generating heat. Right now, if it's a bit warmer outside, it feels hot. Later in the semester, you'll drop those proteins down in number. You won't be generating as much heat. It won't feel as hot. You've adapted to that environment. Does that kind of make sense? Okay. So there's your ATP synthase. Lots of electrons, but I need to get that ATP out in the cytoplasm. I need to get it working. It's going to do all sorts of things. We'll see bits and pieces during the semester, particularly to help enzymes work. So, we'll come back to that video on the end of semester. I've got a few slides to go through to give you an idea of what's going to happen during the semester. You need to write a little bit as we go. I'm going to give you an overview of that. You've got a conceptual view of the mitochondria. That's where all the action takes place. That'll occur just around semester break. But before that, we have to build up, understand what's going on. Instead of saying, here's the textbook, memorise this pathway, because you're going to see an ugly one very soon. Let's think about sitting here, getting close to dinner time. What happens at dinner time? Tea time, or whatever you call it. Food in mouth. What happens to the food in your mouth? Sorry? Okay, let's think about the process. Think simplistically for a start. So there you are. Let's say you have a full set of teeth. What are you doing? Sorry? You're masticating. Masticating. You are breaking it up, increasing surface area, aren't you? Those glands at the back, you see some babies sometimes, they're waiting for food. They squirt out a little bit of what? Saliva. What's in saliva? Amylase. What does amylase do? Breaks up carbohydrates. It doesn't break up fats. There's no other enzymes in the saliva to do that. Doesn't break up amino acids. None do that. Just carbohydrates. Why do you think that's the case? I stick a nice bit of potato in my mouth, a bit of bread. Kind of tastes a little bit sweet, doesn't it? Tastes nice. But only amylase in there. But what is amylase? A protein. Gets to your stomach, pH 1, 2, starts breaking down. We need to add other enzymes later on. So I'm going to take you from ingestion. What happens to that food stuff? We're going to start with amino acids because you just finished FP and G. You should know all the amino acids now, shouldn't you? All the three-letter acronyms, the single-letter acronyms, all their chemical structures. I don't care. What I care about is what their functional groups are, what they do, what their role is. And that's going to be critical for us. We're not going to be testing on that kind of stuff. That's textbook stuff. We have to think about what's going on in you to help make you work. That is, whatever you do, help make you think. But we're going to run through amino acids. We're going to run through fats. We're going to run through carbohydrates. And there's a reason we run through that sequence. Again, very deliberate. We're not going to cover every pathway. It's not possible. We're going to focus mostly on humans, but it'll be related to other species so that you can hopefully relate to it. It makes sense to you. Before I start, my normal sequence is to start the lectures usually on the hour or 10 past the hour. It depends on you. I'm down here early, usually get here about normally half an hour early. So if anybody wants to have a chat, you're welcome. I'll come down a little bit early, no drama. But do we agree to start on the hour and try and finish a little bit early? Or do you want to start at 10 past the hour? On the hour. No, everybody can get here on the hour? So we agree on the hour, okay? So 4 o'clock every Tuesday, isn't it, for you guys? We'll start on the dot, okay? And try and finish it. Now, I usually go for roughly 45, 50 minutes and then have a 10-minute break. Stretch your legs, get something to eat, refresh and get ready for the next 40 or 50 minutes. Questions as we go. Is everyone clear on that? Okay. And there'll be a quiz, and I'll come to that shortly. So we agree to start on the hour. Let's start talking about metabolism. Why does it induce fear and loathing? Well, mostly because I'm teaching it and nobody else is brave enough to. Mostly because it's kind of complex and very conceptual and not kind of fun like everyone thinks it is. But each year we try and modify and enhance some of the things we do. Now, for each slide, I'm going to, like you're doing now, we're going to talk about the slides. Some slides I'll say, stop writing, listen to me. Let's discuss something. And I'm going to say, okay, you write, and then I'll talk. But if you've seen them before, you've got an idea of what's going on the slide, don't you? Why it might be critical. And sometimes you want to write down the things I'm saying. Oh, I didn't write fast enough. Well, just say so. When we get to the end of this slide and I say, everyone finished with this slide? Can we move on? And you'll say, yes, I'll move on. If someone says no, we'll wait. But if we get to the end of the lecture or the end of the semester and you say, I missed some of those slides, can you go back? Is that going to happen? As my dear sister-in-law loves to say with her adult children, NMP. You know what NMP stands for? Must have adult children.
I think so.
What do you think it is?
Not my problem.
Absolutely, not my problem. Why? Because I've given you slides, I've gone through the slides, there are recordings from every campus, there's 3 campuses, I've done it 3 times, I'm overdoing it, no offence, okay? It's up to you to be here to take it on board. Watch the recordings if you've missed something. Don't ask me for those, it's not going to happen. Not my problem. They're there for you already. 3 recordings from 3 campuses, there's no excuse, okay? And I'm busy, I have a number of students this semester who are really keen to get on with some work, so I'm going to be really busy. So what does this slide tell us? There's a seesaw. Anabolism, I'm building something. Catabolism, I'm breaking it up. Anabolism, what did you do? You ingested amino acids, sugars, fats. What else? Nitrogenous bases. I'm not going to focus too much on those, a little bit. But they are just a sugar linked to a backbone, that's all they are. They're also very interesting, as some of my colleagues in plant research are dealing with at the moment. You take them in, you break them up through your digestive system, but you can't absorb a piece of steak into your circulation. You have to break it up into 2, 3 or single amino acids to get it across into your system, and it'll go into the blood. Carbohydrates are the same. You break them up into single sugars or a couple and across into your gut, then circulation. Fats, you want to write down now, you've got to break them up too, but not into single carbons, but into long chain carbons that are activated that go through your gut cells. Does anyone know where they go after they finish going through your gut cell? Through your epithelial gut cell? Do they go to the blood or somewhere else? If you say blood, you know you're going to be wrong, don't you? Where's somewhere else, sorry? Absolutely, lymphatics. But they are packaged up, and I'm going to give you some analogies. They're not just on their own, like you saw in that cell. They're packaged up in vesicles. You know them as HDLs, LDLs, VLDLs. They are literally cargo-carrying lipid droplets. They start in your lymphatics before they get to your bloodstream. What do you do with them? You build things. You built that cell. You built those proteins. But every cell is recycled. Every cell recycles bits and pieces. Some things stay in you forever, almost. There's only one molecule that stays with you the whole of your life as a molecule. I'll come back to it. Every other amino acid, every other protein is recycled. You are a new person in some parts of your gut every week, your bone every 10 or 11 years, your skin every few weeks. You keep changing. That's a good thing, isn't it? Except if you put a tattoo or something on there, okay? When you build something, you need energy. What do you need? Well, I mentioned ATP a moment ago, but there are at least 3 other critical ones to keep in mind. NADH, NADPH, and FADH2. Think of them as electron carriers. Jot it down, electron carriers. So is ATP, really. But what it does, it puts a phosphate on somebody. And let's think about that concept. You've all seen an FP&G, phosphorylated this, phosphorylated that. When you phosphorylate an enzyme, you often activate it. So when it's activated, it will work. That's what ATP does for you. Now, there are some exceptions to that. Sometimes you take the phosphate off and then it's activated. Sometimes you put more on and you'll see how that works. You did some basic Michaelis-Menten kinetics, you know, straight line enzymes, boring as. We're going to see different types of enzymes that kind of get excited or don't get excited. They're not on-off, mostly. They're slow or fast, just like the production line making cars. If there's lots of orders, you make lots of cars. Orders slow down the manufacture. Those are the principal concepts. Anytime you break something, if you burn it, you generate heat. But if you break carbon bonds from these fats, proteins, you can generate electrons and protons. That's what you do to manage it in your cells. Now the balance, this bit here in the middle, is the tricky part. Burning them off, what do you generate? CO2, water and ammonia. Well, you know the CO2 is coming from carbons. Where's the ammonia coming from? Where's the ammonia coming from? Amino acids, absolutely. That's the one thing you can't have hanging around because that's a bit toxic to you. So what do you make? Urea. What do? Off to the porcelain, deliver it to the STP. What's the STP? Sewage treatment plant. Or if you have a septic like me, you live on the land, so to speak. This is a balance. If we can get that general balance, everything else becomes a lot easier. Okay? But concepts become easier. How do we run it? How do we stop it? How do we slow it down? Who are the key regulators? Write that term down, the key regulators. Are we done with this slide? Is that a yes? That's what I need to hear. Oh boy. Now this isn't some made-up circuit diagram, this is real. This real thing is some of the pathways we're going to deal with, each enzyme, each pathway, but we cannot cover all of them. There's no chance in hell, but there's no reason to because it's all online, easy to find out. We need to understand the language of how this thing works. It's like a circuit diagram on any electronic board. If I know how it works, I can get it to do some work for me. I can understand how to fix it when there are issues. Each of these dots and pathways is an enzyme and a cofactor derived from a gene, activated in the cell depending on what the cell's role is. And you'll notice I boxed in red one of the core pathways. You've got to write down carbohydrate metabolism. What are we doing? Well, if you can see, a lot of these pathways feed into this thing going down, going from their carbohydrates down to that circle. What do you think that pathway might be? The circle is the citric acid cycle or the Krebs cycle or TCA cycle, I don't care what you call it. But what's the pathway going down from carbohydrate to there? Glycolysis, good job. Glycolysis. You're taking a 6-carbon sugar or all sorts of carbon molecules feeding into this pathway and you break it in half basically. But breaking it in half, you have to activate it. What do? Put phosphates on. And I want you to jot this down. Anytime you put a phosphate on somebody, you activate it, ready to do some work. That's what phosphate does. It kind of G's them up on some raspberry cordial. You get down to the citric acid cycle, just jot in the middle, that's where you generate electrons and protons. The citric acid cycle's in the mitochondrial matrix. That's where it does its work. Now, these pathways are critical because many people, many individuals, animals and otherwise, have diseases or have adaptive advantages that modify some of these pathways. Now, you might have to mitigate those with drugs, but particularly with metabolism, you can find other ways. People with lipase issues, not producing enough, you change their diet. So metabolism is not just give someone a drug, think about what they need, how they manage it, and that's why metabolism is critical for medicine. If you want to do medicine, if you look online, you'll know you have to do metabolism. If you want to do nutrition, if you want to be a nutritionist, you have to do metabolism. Understand how these things work. Now, as you can see, there's lots of different pathways, and I've mentioned some of the critical ones, didn't I? Amino acids, fats or lipids, carbohydrates. I'm going to start with amino acids. They're as complex as hell. We're going to pick some key examples. How to make simple ones into substrates ready to make energy through glycolysis. Fats we'll come to shortly after. Talk about how you make fat, synthesize fat from sugar, amino acids that have the nitrogen ripped off them, which maybe we should talk about now. Do you store amino acids? Sorry? No, you don't. If you don't use it, you lose it. As simple as that. But fat, that's another story, isn't it? You know you can store that for almost as long as you need to. And the more you put in, the more you can store, which becomes an issue, doesn't it? But that's also an adaptive advantage. Now, when times are tough, you need to store fat. But they ain't so tough in this part of the world right now, are they? Unless you own a property and you're worried about the drought. How you manage the individual as well, it's not just change your diet, as I said earlier, it's how the brain controls it. And the gut produces hormones that control your brain. Oh, I like the taste of that, let's have some more. I want to get bigger and bigger as a stomach. Let's have some more. I'm addicted to food now. I get a nice little run from it. I'm feeling good because of it. Not just in my taste buds, but my stomach says I like that. Those become issues with managing obesity and diabetes, particularly in young children, which we're going to talk about. I'll touch on nucleotides. One, because I did a lot of work on RNA and DNA. But, you know, they're simply sugar, as I said, with a couple of nitrogens hanging around, some phosphates. So once you learn the concepts, well, it's actually easy. But making them is particularly difficult. You don't need to make as many of those because you ingest them, unlike other species. Now, we're going to work around this. Now, unfortunately, I need to convey to you right now, I can't just do amino acids, I can't just do fats, I can't just do carbohydrates, because you can see from this thing, they are all interlinked. So what I'm going to do is start off with amino acids and say, talk about glycolysis generally, how it feeds into glycolysis. I'm going to talk about fats and how they get involved, making them, breaking them down, how it feeds into glycolysis. But I'm not going to talk about the details of glycolysis until later in the semester. So that way, by the time we get mid-semester, you should know glycolysis more broadly better than anything else. Does that kind of make sense? But I'm going to talk about the key regulators. Who's controlling the pathways? How it's working. If I ingest only fat, if I only eat steak, what am I doing to my metabolism? No carbohydrates. Some people do that. I was talking to a colleague this morning. His friend from France only ate proteins because they lived somewhere in the highlands, didn't grow vegetables, no vegetables with a meal. Guess what kind of disease and disorder he has now? That's one of them he did have. Resolved that with some vitamin C tablets. What else? Think about your gut. It's only taking on board proteins. He has colon cancer. Metastatic, probably not going to be here much longer. Understanding the balance in that process is critical as well. So metabolism is not just about torturing undergrads to make my life good, feel better. It's about understanding what affects you. We're done with this one. Can I move to the next one? Okay. This is my timetable and generally yours. The stuff in red is where I'm at. The stuff in black are the labs. You can go to any of those red regions, the lectures I talked about. It doesn't worry me, as long as there's space, which might be a problem at Parramatta, but who cares? That's NMP. You've got 3-hour labs. Some of you still haven't got your labs resolved, which we're trying to resolve. If you haven't, I told you to come and see me. I mentioned that by email. You'll notice if you looked on views, there's my email address. If you looked on views, there's a little footnote saying, I respond to student emails between 8 and 10 weekday mornings. For those of you who've had me before, I do that religiously, don't I? I'm in my office before 8, I respond to emails before 10. After 10 o'clock, I'm too busy. Weekends, they're mine. I don't want to end up in divorce courts, thanks very much. So don't expect a response on Sunday night, midnight. I can't finish my report. NMP. You've had all the time to deal with it. Now, some of these labs are pretty full, as I mentioned earlier. You have lab supervisors on this campus and some of my demonstrators helping out, as my students as demonstrators helping out. They've been through it all. And each year we modify and update and tweak the labs to make them work better for us and for you. So everything's never the same, so be careful if you think you're going to find someone else's lab book and be okay with that. Might be a problemo. Okay, when I say email, I mean email. Don't try and leave me a phone message. Send me an email. 1, I want a record. 2, I don't answer any external calls because I get so many Russian brides, hotel rooms, all those kind of offers. I don't respond to external phone calls unless it's prearranged. Is everyone clear on that? Email. And please note it's M.R.Jones. Jones is a pretty popular name. There's 3 other Joneses Chris Jones might be your DAP for medical science. Graham Jones might be teaching you genes, genomics and human health or whatever. So make sure it's M.R. If you don't put those in, it'll disappear into the ether and I have no idea you've sent it and no one else does. Clear on that? Okay. Can I get to the next one? Here is our schedule. It's up on the learning guide. I put it down below in views. Now this schedule is kind of critical because it tells you how we're tackling the semester. Some of the things you need to keep in mind. I said on views, and you'll notice on views, the front line, unfortunately I'm trying to get the notice in red. Try to do that with IT for a couple of days. They've told me whatever template they've got from whoever the hell that makes that particular software cannot be changed. You know, sometimes you click on it, it might flash up red and then disappear. I can't change. I like big red notices. Trouble is I don't like the red of the university, but that's nothing I can change. But those notices will go up every Thursday afternoon to tell you what you should be focused on the next week. After that week, they'll go into the weekly notices. Is that kind of clear? So you don't have to click on weekly notices to find out what's going on. You only click on those if you've missed a week. Each week there'll be notices up there. Up there, what does it say right now? Lecture material will be available at 1 o'clock. Same thing every week. It says up there you should be watching these videos, training videos online, summarising them in your lab notebook. What does your lab notebook look like? I put an image of it up on views. It needs to be one of these industry standard ones with page numbers already in there, signatures, blah, blah. If it's from another unit, we have no problem. Save a tree. If you get one of these for this unit, you're welcome to use it for other units, including mine. But these are important, not those red and black things with just bits of paper with lines on it. Why? Because this is a lab notebook. In the front, it talks about intellectual property. And coming from CSIRO, that's kind of critical. Because if you reach into your pocket, you'll find some cash, I suspect, which is plastic money. That plastic money that's in your pocket was invented, the polymer was invented by CSIRO, not by me. I was in a different sector. This is intellectual property. That's lawyers telling you how to fill these out. All of my research students have to have one of these. All of my third years have to have one of these. You're going to have one of these because you are doing a project in the lab, developing your own lab manual. So nothing else will suffice. Is that kind of clear? You'll need to get one of these. Now, there's probably a run on these in the Connect bookshop, so we'll be patient until I get those all in, as usual. But that's what they do to us. Why is that important for CSIRO? CSIRO invented that plastic polymer. They didn't have lab notebooks in those days. Somebody else grabbed it, patented it, CSIRO got creamed. Lost millions and possibly billions of dollars because they didn't have a lab notebook. Most organisations I'm aware of now all have lab notebooks. Employers are looking that you can write one of those things up. Write down the details, aim, materials, methods, results, discussion, and be articulate. So you're going to go through that during the semester. So I'm clear on that. One of those. It could be red, green, doesn't matter if it's graph paper or whatever, as long as it's a Griffith Hack industry standard lab notebook. So you watch those videos, just a couple of simple ones. Dilutions, pipetting, summarise them in that lab notebook. Lots of things you're going to write in there. Some things you can just stick in or slip in the back. The supervisors will advise you of that. So we're doing an overview. Overview, there it is. Overview of the unit. In previous years, we'd have a briefing in the lab, but because of so many students and not enough space to run all these labs, the briefing's gone. So next week, I'll come back to this in a bit of detail, you have to be ready to go. You have to have those summaries done, read the material I'm showing you today, and have your lab notebook ready to roll. Now, you work on this campus in teams of 3, mostly 4. You're going to be a team leader. All of you are going to be a team leader at some point, unless you do a runner. You're going to be a team leader. You have to manage a team of 3 people, and that part of the project, you'll be a team leader for Module 1, Module 2, Module 3, or Module 4. You're going to be developing an assay from first principles, all of the key bits you have to work through, what an employer is looking for, how you problem solve. That's what they're looking for, how you articulate that. If you look at the learning guide, you'll see point 7. Let's get to point 7, shall we? I didn't put that thing up because it's a bit big and ugly. That is on the slide. It's on views. And as usual, our administration does a few screw-ups here and there, but I can tell you, point 7 right here. Keep this in mind all through the lab. Demonstrate competency in time management. Time is money. Through identifying milestones and reporting progress against those objectives. I'm going to give you a project outline with a series of milestones and objectives that you do each week for each of these modules. In accordance with human resource management and industry expectations, the feds are driving us to make sure you're job ready. It's a bit hard in metabolism because, you know, there's a lot of conceptual stuff going on here, but we can still do it. Why? That's probably why I got the job to deal with metabolism, from where I come from. So you're going to have to manage those processes. You have to be prepared. You will be a team leader, and I'll nominate those as soon as I get the final class list on Monday, at least as close as I can get. I'm going to nominate a team leader for a team of 4. You'll be nominated into that team. I don't care who your friends are. That doesn't count, because when you get a job, do you work with your mates? Not usually, unless you're a bit kind of incestuous in that sense. Your job is to manage those 3 people that you're dealing with. What are you doing? Well, in Module 1, everybody needs to bring in a beverage, a solution derived from some plant. Might want to jot it down now. A plant-derived solution. Could be juice, but as long as it's something we don't have to centrifuge, filter, isolate, it's in solution. But what you should have, write it down, is a nutrition label. Okay, a nutrition label, so you know what's in there. You're going to be developing a protein assay to measure the protein in solutions, in samples, at least for Module 1 through 4. First one is a plant-based solution, a beverage. Everybody brings one in. Now, you might bring one to the lab and think, well, you know, I want to drink this. But as you can imagine, you can't take it in the lab. The lab's managers, I've asked them to have sample vials, so you can pour in an aliquot into that sample vial, leave your juice or whatever it is outside, drink that later, bring the sample in. You only need a couple of mils at the most to do your particular project activity. Kind of clear on that? But make sure you've got the details of your nutrition label. In your team of 3, you're going to get in the lab a red dye. I'll come to that a bit later as well, that detects proteins. But we need to know what kind of proteins, what kind of amino acids. In the lab, you'll have 20 amino acids in solution and this red dye. You'll have a BSA, write it down, bovine serum albumin. BSA standard solution. That'll be at a known concentration for you. The first lab, that'll be the concentration ready to go. After that, it might be any concentration. You'll have to make dilutions. Be aware of that. Supervisor reminds you. You'll have red dye, BSA. What's BSA? Bovine serum albumin. I'm going to talk a lot about it during the semester. It's in your blood. It carries all sorts of things in your blood. Hormones, things like steroids, cortisol, testosterone, other small molecules. It's around 50 kilodaltons, very ubiquitous protein. What's a bovine? Cow. We could use HSA, human serum albumin, or chicken albumin. Bit of a problem with humans, but we use cow. It's nice and cheap. But it's a standard, a known concentration. What about your beverage? Do you know how much protein's in there? Well, the nutrition label might say, but you're going to try and work up an assay. The first part of that is you're working up specificity. You're going to have 20 amino acids. There's 4 of you. You're going to do all 20 amino acids? Well, think about how you manage your time. And this is critical for all of you. How many hours have you got in the lab? 3. You're going to use a 96 well plate. You've all used one of those, haven't you? 96 wells. There's 4 of you. How many wells each, roughly? A couple of dozen. You need a standard curve, don't you? Probably. The team leader manages the 96 well plate. Everybody else will get tubes. Make all your dilutions, do what you need to do. Put all your samples on the 96 well plate. The team leader goes to the plate reader. You all used one of those, haven't you? Gets the data, photographs it, hands it to his colleagues. Okay, let's deal with it. What's happened? What have you done? How'd you do this? But you had 3 hours. How long does it take to read the plate? Let's go back further. How long does it take to distribute the data to your colleagues and discuss it? What would you guess? Sorry?
5 minutes.
That's probably a bit tight. Particularly, remember, this is the first time you've done it. Now, this is kind of important too, because as we'd say in advanced cell, if it's the first time you've done it, are you wiser to give yourself a bit more time rather than trying to keep it really tight? When you would know what you're doing, then you can tighten up the time frame. So roughly how long do you think? A bit more than 5? 3. If you take 3 hours, you're going to get 0. But it doesn't take 3 hours. What do you reckon? 10, 15 minutes? Okay. Okay, I've got data to my colleagues. Now I'm going to measure the plate. How long does that take? Zit, 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 zit. A couple of minutes. But what if there's a queue? There's 2 plate readers in the lab, 40 plus students in each lab, 10 team leaders, 10 plates to be read. How long would that take roughly, do you reckon? I'd give myself 10, 15, 20 minutes. Okay, let's work backwards again. How long would it take everybody in a team of 4 to put all their aliquots onto that 96 well plate? And if they're good at pipetting, it might be really quick, mightn't it? If they're not, you know what's going to happen, don't you? Because there's people in this class who didn't do all those things earlier, so it's going to, how do I use a pipetter? How do I do that? That's a team leader's responsibility to make sure that happens. How long would you give yourself for putting all those samples on? 10 minutes. Ooh, that's a bit tight, isn't it? If you were good, I'd say 10 minutes. I'd give yourself half an hour at least for a start. Remember, it's the first time you've done it. You don't know who your team members are. Can they pipette? Do they know how to use one? You're supposed to have a licence, but I have to say many times I can't see, if you drove like that, we'd all be dead, wouldn't we? Give yourself half an hour. Okay, making up your solutions and dilutions. How long is that going to take? Ooh, that's going to hurt, isn't it? Yeah. Now go back one further. I've just brought in my samples. Here I am, the team leader. Here's 3 people I've never seen before. I'm Mark, and you're, what are we going to do? I know that takes, what we say, 15 minutes, another half an hour, another half an hour, another half an hour. That's an hour and a half. So we've got an hour and a half to sort ourselves out, don't we? What we're going to do with our sample. There's the beverage. You've got juice. You've got juice. You've got juice. They're all different juices. I've got a juice. Who's going to make the standard curve with BSA? Team leader's responsibility. What do you do with your samples? Well, I want to measure protein, but it says this much on the nutrition label. How do I know it's going to work in there? Oh, there's the 20 amino acids. I need to know specificity. Who's going to do what amino acid? Which one are you going to do? 20 of them last semester. Should I do families of them? Should I do different family members? So maybe the hydrophobic, the hydrophilic, the aromatics. Or should I do all of those one by one, 20 of them on a plate? What concentration should I use? Now, this is like baking a cake. Has anybody baked a cake? Yeah? Got a recipe, haven't you? But think about it. If you walked into a kitchen, you'd never seen a recipe before. There's the oven, there's the pan, there's the flour, the egg, there's the mixer. What do I do? Well, look for somebody's recipe to give you a head start. Otherwise, you make lots of mistakes, don't you? But that's what we want. We want you to make mistakes and learn from them, because you're learning how science works. You're learning how to problem solve. You're learning particularly that lots of this stuff takes time. It takes problem solving. To help you, which I'm not that comfortable doing, as you can imagine, this thing's going to appear. I'm going to take you through one today. What is this? Write down WIL, a WIL assay, a work integrated assay. This one's urea. Sorry about that. This is urea. You're going to need that. Where does it say there? Workshop 1, will urea assay. This will be available to you. That is the catalogue number from the supplier. You'll have to go and get it, just like we do in the real world. You'll have to print this out, read it. What does it say? They did this, this and this. Here's some background. Oh, it relates to the lecture material. That's funny. Curious how this Jones does that. They do this, this, and with their samples. This is the volume we're using. Just please note absorbance assays. Fluorescence assays we're doing in the advanced cell unit, not this one. They're using a plate reader like you, a 96 well plate. They did this, this, and this. They waited this time to measure urea. Can I use that as a template to start how to cook my cake, except I'm making a protein assay? Why not? Tells you roughly the volumes, tells you what they did for a 96 well plate. Yes, it's urea, it's a different assay, but you will discover towards the end of the semester that all assays are similar. They do similar things. It's just what you're trying to measure, understand those concepts. So this is going to be available to you. It'll be available till 1 o'clock this Thursday. Click on it, get the one you're going to use, this urea one, and start to get an idea of it. The beauty of these companies, like Sigma, for example, on the back they'll put up, frequently asked questions, problem solving. Now that's critical because when the team leader gives their presentation, you're going to be asked these kind of questions. Not just about the urea, about your assay. What's going on in your assay? How did you solve that problem? Now right now you think, oh my God, what am I doing in this unit? And that's a good thing, because if we have less students in the labs, it'll be much easier. There's plenty of space at Hawkesbury. This thing also tells you average samples, standard curve. I mean, it's a giveaway. And you'll get a different one of these for each module. You see up there, you're going to do a lipid assay for module 2. That matches up with the lecture materials. But lipids are measured differently. They don't measure all the carbons, they measure the free acid on the end. Then we're going to do this one down here, PP. What the hell's PP, not NMP? PP is related to glycolysis, phosphoenol pyruvate. Still have to measure that, similar assay, similar issues. And then the mid-semester exam. Why is that there? Well, for you, it's kind of lucky again. I had my arm twisted, almost broken, to have it online, take home, available from the Tuesday through to the Friday, Tuesday 9 a.m, Friday 5 p.m, to do at home and collude as much as you want. I'll come to that later on. But you know me, already thinking, what a jerk. Questions aren't going to be so straightforward. You'll have to think about them. But they're all multiple choice. That's a good thing. And everyone asked me at the end of the semester, all these questions and answers, can you give me the answer for the multiple choice? I did. It's in front of you, one of the 4 that I've given you, more than likely. Which one? You'll have to find out. Then we have the break. Module 4. Oh, sorry, I need to back up a tad. Module 1, you bring in a plant beverage. Okay, clear on that? That's done, dusted. Module 2, you're going to bring in an animal-based beverage. Milk, fish sauce, anything that has a nutrition label derived from some animal. Clear on that? Everybody brings one in, including the team leader. Different team leader, might be a different team, depends on the supervisors. Module 3, you're going to bring in a manufactured beverage or solution, something that's not derived from animals or plants, like Gatorade stuff or Coca-Cola or whatever you want, something with colour. Because that lab, you're going to run the full spectrum of your red dye and the beverage or solution you bring in. Because the solution, like red Gatorade-y stuff, whatever they call it, mother, might interfere with your assay. What do you know? You're dealing with interference, things that might screw up your assay. Questions, queries? If you're asking why you're here, you're either forced to or you want to. If you want to, check with your GP, you don't need to take some pills. If you're forced to, suck it up. Oh, you might need more medication. Yeah. All right, module 4. You're dealing with the range. You start at the beginning, how specific it is. The next one, you're dealing with sensitivity, how low, trust you, how low can you go? What's the lowest amount you can measure for the standard and your sample? The next one's interference. There it is there with your synthetic or manufactured beverage. After semester break, after you've survived the mid-semester exam, we're going to supply some urine. It'll be fresh because if it sits around for a while, it's pretty much on the nose. But that urine will be from different patients with different issues. Some of those issues might be too much booze, too much water, too much protein, excreting red blood cells, some other issues with their blood. How much sample, what dilution do I need to make it? What's the dynamic range of the standard curve? How far can I go to a high concentration before that thing flattens off? Can I get an accurate measurement? That's critical for the urine. You get down here, we get an NADH assay. That relates to the lecture material. Remember that? Key electron carrier, NADH, one of my favourite molecules. Why is my favourite? I'm glad you asked. Because it's one of the few that fluoresces in your cells, and that's the work we do. We track it in the nucleus because it's involved in stem cell differentiation in the first 4 hours of your life and every stem cell. We get down to week 12, what are you going to do there? There'll be some more instructional videos. Those videos are for ELISAs. Anyone know what an ELISA is? Not someone you date or someone's best friend. ELISA, what is it? Sorry? Kind of, yeah. It's an enzyme tag. It's called enzyme-linked immunosorbent assay. It uses antibodies, and I'll talk about that as we go, with an enzyme on it that generates colour. Now that standard curve is not nice and straight like you're going to be used to all semester. That's going to be that ugly word, a log curve. Okay? It's got a huge log curve. You need to watch those instructional videos. Then you will come into Module 5. Module 5, we're going to give you saliva samples from patients. Now keep in mind, Module 1 to 4, you'll be in teams of 4, right? 1 to 4, teams of 4. Module 5, you can work with whatever the hell you want as a pair. But Module 5 is a test, a skills test. Can you pipette? Did you watch the videos? Do you understand standard curves? The skills test is going to ask you, can you measure the concentration of cortisol in that saliva? That's what the instructional videos are. I'm going to take you through some of that in the lectures as well to help do that calculation. But there's another thing you have to do. You've been doing protein assays all semester. How much protein is in the saliva? We know. You have to find out. It's what we call a quality check. But also there's quality controls that we run in those assays, and you'll see those in the videos. And these will become available as we go through the semester. So you're going to do a cortisol and a protein ELISA, bless you, for Module 5. Module 5, you'll do that over 3 hours. Carefully designed. Please note, the protein assay you do all by yourself. There's your sample, your own standard curve, put on a plate, measure it. The cortisol assay, the demonstrator will do the standard curve. Why? Because they're expensive kits. You'll get 2 wells of a 96 well plate. They come in strips of 8, pluck them out, break them in half. 2 wells for you, 2 wells for me. We manage it. Once those wells are in your hot little hands, the acronym is NMP. The demonstrator will tell you, do this now, wash now, add this now, do that now, read the plate. Here's your results, off you go. You'll do your calculations, you can do them in the lab. You will have an hour after the lab session ends to go online and submit your calculations, your value for cortisol and protein onto Views, because you'll get a patient number unique for you. Is that kind of clear? I'll remind you of all this as we go through the semesters. The supervisors will as well. So we'll keep coming back to it. Once you're finished, you go home. There's no lab book marking, and I'll come to lab book marking shortly. Questions or queries? Okay. Lectures, I'm matching up what we're doing in the lab all through the semester. Just note the first half, we're inside the cell. Yes, we ingest, but how do we get it in the cell? What does it do in the cell? It goes from cell to cell. After semester break, it's integration. How do you manage it? What affects you? Well, that's why you're doing a cortisol assay, because cortisol affects metabolism. We're going to measure in saliva, a nice non-invasive way to see if your cortisol levels are up, changes your metabolism. What's cortisol? It's a steroid. What do steroids do? They change gene expression. They're called transcriptional regulators. They're going to up or down regulate things. Cortisol does both. It's a nasty piece of work. It can make you more obese or less, depending on what it's doing in your body, how you feel. And that's what we focus on the last half of the semester. Fortunately, the textbook's finally catching up. And you'll see in the morning, before you get up, your body's already priming, sometimes I wonder with students, for muscle activity by activating pathways ready to do work. Again, I wonder with students, particularly my own daughter. That means your metabolism is priming for each day's work. That's what happens to you. What else we got? Clear on the sessions. The sequence while we're doing it. Yep. Can I move to the next one? Cool. All right, the first thing you have to do. Up on views, there's this risk assessment form. You need to fill this out before you can come into the lab. They won't let you in unless you show this to them in handwritten blue ink. It's up on views, and you need to fill it in for phosphoric acid, 1 molar, and methanol, 100%. It's not rocket science, not like the kind of risk assessments we have to do, but you need to do that pronto. The other thing you need to do, and you've all done this already, haven't you? You know that work health safety online thing? I have to do them too. The certificate that says you've done them, you need to flash that to the supervisor as well. Another unit last semester, it's fine, you just show that to them. But please make sure you have this one. You'll need to do another one of these when we do the ELISA later in the semester, different molecules. Now, you are a team leader. You will be at some point, so you have to manage time. If you're late starting, if you're late finishing, and it's not our fault, you will be penalised, so will your team. Time is money, as I said, and indeed it is for us. If our supervisors, demonstrators have to go over time because you're too slow, I have to find money to pay for them. I have to go on my hands and knees to the school and say, can I have some more money? Why? First of all, why? Why is it going over time? What's going on? Oh, they're a bit slow. That's not an excuse. Can I move to the next one? No? See, I'll wait. Okay, now I'm going. Keep it simple in these boxes. Okay? I don't need war and peace. Why do we need this? Because hopefully you realise from the things you've already done, legally, you are responsible for what goes on in the lab as well as we are. So if you don't know what you're dealing with and you start messing around, you can be legally liable as well. So you must behave yourself. You must understand. These labs I've cleaned up over many years, so there's not too many nasties, and they're not as nasty as they used to be. Because some of the labs were pretty awful, I have to say. But now we're focusing on different things, particularly problem solving. That's critical for us. Can I move to the next one now? Is everybody else right? Okay. Here is the sequence, and I've already touched on these, so I'm not going to spend too long on them, but think about the first module is specificity. You have 20 amino acids. Can you coordinate with your colleagues around the lab to see whether they pick up different amino acids? That's up to you. The supervisor has been advised to guide you, not give you the answers, guide you to the answers. Should you do this? Should you do that? Do we know the answer? Depends on what you do. And what I will say to you while you're just sitting there thinking, I should write all this down, we don't mind if you make mistakes, but your lab book should record everything you do. Your lab book, so that if we went back and did what you did in your lab book, like baking the cake, we could do the same things, even make the same mistakes. That's fine. But if you don't write it down, you'll get a nice round number. So write down what you do. So if you say, this result's not what I expected, That's a result. A lot of research students don't realise that. That's what you get. That's how it is, because of what you did. Not because of you. Yes, you might be slow preparing, not that good, but you'll get better at it. That's why you're doing it a few times. The first one is specificity. Please read this document. It'll be available at 1 o'clock on Thursday. It's the milestones and objectives. I'm going to touch on some of those in a moment. The first one is about specificity. Will the assay detect only the desired molecule? What are you trying to detect? Proteins. Here's all the amino acids. Does it detect those? Which one? Does it matter what concentration for a start? That's your decision. But justify why you're doing that. And it won't be right or wrong, but use that template, the urea one, as a guide. The next one is sensitivity. Will the assay detect the concentration of the molecule that you're sampling? Well, you're bringing in an animal-based beverage for that one. Often milk has lots of fat in it, doesn't it? Is that going to screw up your assay? Is there much protein in some milk? Well, it depends on what they do to it, doesn't it? Or other solutions. The next one, interference. Now, there's that synthetic beverage. There's Gatorade screwing up your wavelength of absorbance. How much? Can you do something else? You're going to run a spectrum to see, can I go some other wavelength, for example? The next one, range. Here I am, I've done the low bit, the specific bit. How far can I go up accurately before, oh, it's no good anymore? I can accurately measure. The last one is the quality control, which is a test. Can you measure it? But quality control on each assay, about who does it, the reagents, where it's done, the equipment, all those things. We done with this one? Okay. This is the marking rubric for your lab book. Everybody's going to have their lab book marked for Module 1, 2, 3 and 4. There's no lab book marking for Module 5. We don't need to see your lab books. You do whatever you want. It's up to you. That's a test, skills test. First of all, you're going to get what we call binary marking. It's either 0 or 1, nothing in between. First thing is the title and aim accurate? Well, if you look at my objectives and milestones, it says what you should achieve for Module 1. Is that the title? What's the beverage you use? Is your beverage, your solution in that title, in the aim? If it's not, is it accurate? Nope. That's a problemo, isn't it? So what are the supervisor or demonstrator marking going to give you? Nice round number. Be accurate. Be specific. The materials, I've got a list of those already to give you at 1 o'clock. But have you got your beverage? Which ones did you use? Which amino acids? Not all 20. If you used all 20, say all 20. If you use some, say some. What concentrations? You must say. Is that kind of clear? The methods, how did you do it? Well, if you have to bake a cake and you're trying to put that methodology on the back of the packet to sell lots of cake packets, it should be sequential, shouldn't it? Methodological. If I'm doing the same thing I did last time, then I can just say refer back to page so-and-so. Don't make life difficult by rewriting the same things. That's pointless. That's why you're using those lab books. Refer to page so-and-so for your methodology. Your data and results. Well, you've got a photograph. Now you need to summarise them. Great. There's one less. You need to summarise your results, but put your raw data in. We need to see your raw data. Is that clear? Not just summarised data, your raw data, and what you do with each step in that data. How you manage it. As long as it's logical and legible, we don't have to worry. Make sure you have the right, bless you, significant figures, appropriate units. We kill students for not having the right units of measure. A number on an axis means nothing unless it says what kind of number it is. Absorbance units or concentration, mg per mil, whatever it is. But if it's not there, you automatically get a nice round number. Make sure you have legends and titles on there as well for each graph. Looking at a graph, what is it? I don't know. What have you done? What have you found? How you interpret it as best you can. Okay? Please note that as best you can. Look at the model we've given you, the exemplar, the urea assay for a start. What did they do? How did they interpret it? Was it low? Was it high? And keep it relatively simple. 2 down. Okay, we're almost there. It needs to be relatively well laid out. It should be sequential and logical. But you see, you can lose a mark on each of those, but if you do it right and simply, you'll get a mark for each of those. Yeah? Straightforward. They will mark your lab books in the workshop. So if we go back a slide or two. So workshop, doors open up, come in, lab books on the bench. Some of you will peel off to the side, take a seat, demonstrator will grab the books and start marking. Is that clear? So you'll have from the wet week through to the start of the next session to update your lab books. As soon as you come in, we're going to start marking. Yep. Yes, ma'am.
And maybe we're going to get to this. What do we do in that second workshop week?
I'm getting to that, but that's okay. Part of that is marking your books, but also the team leader, which I'll get to shortly. Now, this is worth 3% for each of module 1 to 4, okay? Everybody's book, including team leaders, will be assessed each week for module 4. Is that kind of clear? Yep. So your beverage, solution, plant-based, animal-based, what else have you got? Manufactured, and the last one? Urine. Okay, we're done with this one. Is that a yes? Yep. Can I move to the next one? Cool. Okay, the other thing you're going to do in that workshop is your team leader is going to get up with one PowerPoint slide. They will need to submit that slide, that is all of you are going to be a team leader, 2 views via Turnitin before the session starts. You'll have until, if you've got a 9 o'clock start, till 9 a.m. to submit the views. You come into that session, bring in a memory stick with your PowerPoint slide on it if you're a team leader. Supervisor will say, okay, who's going first? PowerPoint slide in the lab, particularly on this campus. If you're in Parramatta on Tuesday, you have to go to another room, but that's a different story. Team leader gets up and talks about, shows their PowerPoint slide, what they did with their amino acids, their beverage, their solution, their standard curve, some raw data summarised on one slide. Is that clear? We don't need references on there because it's your work. You can put some on if you want, but don't have a second slide. That PowerPoint slide should be clear, should be logical, it should be visually stimulating. It shouldn't be kind of like a piece of A4 page like this thing I flashed to you before, because that's not a PowerPoint presentation, is it? No. Make it like a PowerPoint, like we do in the real world. We summarise the information. We might want to show some raw data. Raw data's in your lab books. Summarise what you've done. Those team leaders will have minimum 3 minutes. If you finish before 3 minutes, what do you get? Nice round number. 5 minutes, the supervisor will say that's 5 minutes. If you don't finish in like 30 seconds, finish whatever's you're milling around your mouth or your head, get a nice round number. Stick to time. Now, we're not assessing your delivery, whether you're shaking at the knees, whether you're foaming at the mouth, whether you're really nervous, you stumble on words. We're not assessing that. We're trying to get you to practice a graduate attribute, which is communication. First your PowerPoint slide, originally your lab book, then your PowerPoint slide, and then the oral delivery. The more of the practice, the better you'll get. We're not assessing that, but we are assessing whether you can summarise your slide, summarise your data. Again, it's the same kind of things we saw in the lab book. Is the slide appropriate? Decimal place, layout, that kind of stuff, logical, and did it hit the milestones? The supervisor will mark that. They'll give you a 0 or 1. Is it there or is it not there? Is it on time or not on time? That's worth 5%. You need to think carefully about what you're going to say. What you're also going to get in those workshops, so those presentations, based on my calculations, will take about an hour and a half. That leaves us an hour and a half. Lab books are all marked. The scan sheet, this thing here, which is on views, and this one, usually one sheet, will be assessed. We'll keep that. We'll scan it. They send a copy to me. They'll give it back to you the following week. Okay? Oops. This particular sheet also points out down below this will assay. Your first will assay template model is the urea assay. In the lab, the supervisor will give you a patient number and say, here's your patient number. Here's a team of 4. We all have our own patient number. It's the team leader's responsibility to help do the calculations for urea. Calculate the urea concentration for all 4 patients. Develop a standard curve, equation of a line. Calculate the urea concentration for all 4 patients. Team leader comes back up. Here's our standard curve. Here's our patient values for each of us. 1 or 0. Did you get them right? Or did you get them wrong? Or at least 1 wrong? 0. You work as a team to do those calculations. Why do we do that? Well, because you don't do the kinds of things you do in other units where it's on your own, you can't work it out. You work as a team. You can go and talk to somebody else. Supervisor and demonstrator are going to guide you again. But you need to work through exactly what this thing tells you, because this thing tells you exactly what to do. And one of the key ones I want you to write down now is that in a 96 well plate, which is what they're using, you have a blank, right? You've all had blanks, haven't you? Blank moment, blank well. A blank is what in a well? In a 96 well plate. What is it? Yeah, essentially it's a control, but you can have different types of control, positive, negative controls. If I look at that well that has nothing in it, or maybe just water, as a blank, and I get a value, an OD value, let's say it's 0.04, for example. That's pretty common. And I have another replicate, it's 0.042. What have I got? The blank value for those 2 wells. But shouldn't every other well have the same blank value no matter what I do to it? Yes. Every year students get caught on this in this unit. What do? Well, you're used to averaging those blanks, aren't you? Here's my sample, standards, whatever. What do? Average those. What do you normally do? Subtract the blank from the average. You should not do that. Why? Because you're already compounding error. The blank wells, yes, you average them, but you subtract that blank, jot it down now, from every sample before you do anything to it. So that when you start averaging, you've already removed the blank value. Does that make sense? That's critical. Every year someone gets caught out, and I have to say someone, lots of students, because they're just on that borderline and they've got the rounding wrong because of what they've done. The other issue of rounding is Excel. What does Excel do? Which we want you to use Excel, bring computers into the lab, whatever. What does Excel do? How many decimal places does it go to? 9, 10. It shows you only 1, 2 or 3, whatever you say, but reality, when you look at it, it's all in the background. Make sure you round to the appropriate decimal for your work. How many decimal places does this plate reader get to? 3 or 4 maximum. If Excel's got 9 or 10 decimal places, that can screw you up big time, can't it? It'll catch you out, and I'll catch some out, guaranteed. Make sure you focus on what Excel does. So that's your presentation. Team leader responsible for the team, what they do, how they do it, when they do it, and the will calculation. First of all, it's urea. The next will calculation will be fats. Different team leader, fat calculation. Last one, or second one, will be what? PP. Last one will be NADH. But everybody acts as a team leader. You should all be getting better at this much faster, shouldn't you? You should all be getting full marks for it, because it's not rocket science, is it? It's just being methodical. But listen to your team leader. Guide them, as we'll guide you as well. If you don't trust your team leader, talk to others. We're not going to stop you. It's not an invigilated exam. It's about how you resolve problems. What's your problem? The calculation. But be aware I put in a few little dilution factors and other things to make life a little bit challenging, as you can imagine. Comfortable with this one? Does that answer your question?
Yeah.
Okay. Yes, ma'am.
When did they start? So do they start next prac? So do we have to put the first PowerPoint next prac?
So next week you're in the lab getting your data. Okay. The week after, so if you're a team leader next week, you'll have to present in week 3. Okay, so think of the presentations are in the workshops, the dry lab. There's no presentations in the wet lab, which is the grey process. So you get all your data in the grey lab, the dry lab is the workshop. Good point, to make sure everyone's clear. Can I move to the next one? Oops, are we clear on this one? Any other queries? Sorry? Which one, man? That one?
Not next one?
That one?
No, next.
That one?
The slide that you were talking about.
This last one?
Yeah.
Yes. Okay, we're there. Okay. Okay. So your PowerPoint slide should be submitted to turn it in before the session starts. If it's a 2 o'clock start or whatever, 2 o'clock is the last time it needs to be submitted. But still bring it in PowerPoint on the stick so they can plug it in and get going as quickly as they can. They've been told to give you at least an hour and a half to do the will calculations, but you might need a computer. We're going to ask you to plot it out by hand, just give you a rough draft, but use Excel. But don't get tripped up by Excel. We done now? Yep. Everybody else right? Can I move to the next one? Okay. Here's the first assay. It's not urea, because you're going to read that one. This is alanine. And I just want to show you a couple of key things on this before we go on. In red, that's the catalogue number. I'm going to put that on views for urea. You need to go to who? The supplier, Sigma, and go to that catalogue number and get that assay. The urea one I was flashing around you couldn't see well, which is for a reason. But get all the other information related to it. You might find standard curves are not printed like this one. This one I've kept simple for you. It's on the actual protocol. Some of the others are not on the protocol. Make sure you get the details. You might want to find some other publications to help you. So they have that. These companies are trying to sell things to make life easier. So make sure you get that information. This protocol, normally they give you a background, which you'll see relates to the lecture material. And I'm going to talk about these in the lectures. So next week I'm going to be talking about urea and the urea assay. You should be thinking about what it does. Gives you a bit of background. What's happening? Well, this particular molecule, alanine, as we're going to see next week or so, is involved in the alanine cycle. What's that? What's alanine? Do we recall what alanine is as a molecule? How many carbons? Yeah, 3. 3 carbons. How many carbons in a base sugar? Glucose. 6. Okay, you hear them talking about the alanine cycle. Didn't we say earlier you don't store protein? What do? If I don't use it for that down the gym in the 6 or 12 pack that I've got, you lose it. Where does it go? Amino acids back via the alanine cycle, back to the liver. What does it do? Rips off the nitrogen, sends it down to the STP. What happens to the alanine? Starts making fat, but it can also be turned into glucose. Going back up that pathway, let's go all the way back to it. Back up this pathway called gluconeogenesis. Your liver's job is to make glucose for your brain. This thing doesn't use fat. It's full of it, but it doesn't use fat for energy. It only uses glucose and a couple of ketone bodies. This thing will use glucose and fat. Fat particularly it's good at. This has got to have glucose. If this doesn't have glucose, that's it for you. So it gives you some background. It tells you here what wavelengths to use. What should you use? You're doing a protein assay. Well, the supervisors are going to give you a wavelength to start with, at least to give you a pointer. But I said before, we're doing absorbance, not fluorescence. That's for the 3rd year unit, because it has other issues in fluorescence we have to manage. Tells you what's in the kit, all the bits and pieces. There's my bits and pieces on the lab bench. They'll tell you where that is. Tells you how they'll last, what they do, what to manage in terms of precautions, how to store things. If I don't store it correctly, it breaks down, I've got no assay. I've just wasted my money. And indeed, those cortisol ELISA kits are incredibly expensive. The school squeals every year that I spend too much money on that. But you only get 2 wells, so it's actually pretty cheap. What else does it do? Alanine, converting enzyme. It's using a particular volume, 220 microlitres. Now, I just want you to jot down one key thing that we're going to really hammer you on. When you put in units of measure, you've seen microlitre, like a mu. Yep, mu, your way. If you put a U there, doesn't count. Excel does that to us all the time. You need to use the appropriate term in this case, the mu, the microliter, micromolar, whatever it is. Make sure they're appropriate. So that gives you an idea of what they're up to. What did they do? What's their protocol? Now this is where the team leader's got to be involved. Who's going to be team leader? Well, you won't know for a little while. So you want to be reading these protocols. Let's read it. Do you know what's going on here? That's what I store. There's my samples. There's my concentration of my alanine standard. What is it? 100 millimolar or 100 nanomoles per litre. We're going to vary things up to make your life more challenging, to do calculations more confidently. What do? They store the kit and they're going to do the colorimetric, not the fluorimetric. So just draw out fluorimetric. We're not doing that at all. Colorimetric or absorbance assay. What do? They take 10 microlitres of the standard, the 100 millimolar of that standard, and dilute that into 990 microlitres. How much is 990 microlitres? It's about a mil. How much volume can you stick in the well of a 96 well plate? How much do you reckon? A 96 well plate, think about it. 300, it starts to overflow. 300 microlitres is overflowing. You don't want that much. The volume's up to you. But if it's less than 100 microlitres, it might be a bit of an issue, mightn't it? They're going to tell you roughly the volume. But you're going to make up your dilutions in tubes before you put it on the plate. So what you use is up to you, but the bigger the volume, the more the cost, and we'll send you the bill. So think about being prudent with what you've got. Can I measure out 9.9 microlitres and 1 microlitre of my standard? If you're a gene jockey and you're really good, yes, but not very accurately. So you've got to find that balance of accuracy, which is what you're trying to do. They mix that in water and they make up a series of standards. How many standards have they made up? From this, 0, no standard, 2, 4, 6, 8, 10. How many does that give us overall? 4 or 5. When you get your BSA, how many standards should you make up across the range? We just do serial dilutions, for example. The videos you have to watch and summarise. Should I make up a dozen? They didn't. Why should you? Make life easier. Should you have replicates? You've done enough assays to know. The answer is say yes, don't scare me. Okay, say yes. Yes, you have replicates. Skip the fluorescence. What do now? They take their sample. Do you know how much protein's in your beverage, your plant-based beverage for a start? The nutrition label will give you a hint, maybe. But if you had juice that had pulp in it, like orange juice, there's a lot of chunky stuff floating around there, isn't there? It might vary from season to season. Should you trust that nutrition label? I wouldn't. Taught nutrition students biochemistry for a while. They realised that's dangerous. But they're going to do a similar assay to you. So they might look at an average value across a season, for example, or across different regions they get their juice. And they don't do it, they get someone else to do it, like you, to go and do it. So think about what dilutions should you make your sample. Should I make half a dozen, 15, 20, 100? Or should I think about something like high, medium, low to start with? Would that be wise? Yeah, of course it is. Be prudent. A 96 well plate, 4 of you, we've roughly got 2 dozen wells each. If you halve that, because you're using replicates, it gives you a dozen wells to play with, doesn't it? That's not a lot, really. Who's making up the standard curve on the BSA? Team leader. You need to work out how that's going to happen. The first assay, you've got amino acids. How many are you going to run? What dilutions? That's up to you. There's no right or wrong. Please be aware, there's no right or wrong. It's just your methodical approach, because you have to interpret your results at the end. That's science. Your problem solving. They left it for a certain period of time. Do you monitor what's going on? Should I put it in the dark? Should I watch the colour change? What happened? Did it foam? Did it bubble? What's the volume in there? Should I keep the volume the same in every well? You've done enough assays in microplates and cuvettes to know, probably. Does it matter what volume? Absolutely. Well, to some degree, but as long as they're constant, yeah. There's my data coming off my plate reader. What wavelength do I use? Are you centrifuging anything? I said no, because you're using solutions. If there's chunky bits in there, try and avoid those. That's your base assay. They tell you how to do the measurements, the calculations. That's for alanine. The first one you're going to do is urea, isn't it? But you still have to do it on your samples. Okay. Think about what's going on in the well, in the tube. Do I need an appropriate ratio? What is appropriate? 10 microliters of my sample, 990 of something else? Well, that's what they did. That'd be a good place to start. But if you can try something else, that's totally up to you. 50-50, 100 to 1, doesn't matter. It's up to you, as long as you justify it. And I encourage you to experiment. Yes, you have limited time. Yes, you have limited resources. But try a few things. Find out for yourself, for your samples. We're done with this one? Okay. The back of this alanine gives you a standard curve. People always ask, let's get Excel to plot that standard curve. Are they replicates of samples? Yes. What do you do with outliers? Well, we want to see your raw data, and you're going to summarise them, but you need to justify why you might remove an outlier. Either it was foaming, or somebody didn't pick correctly, whatever it is. And don't say operator error. Tired of hearing that. Well, what have I got? An axis down the bottom, nanomoles of my standard. Should I run my beverage as a standard? My sample, my plant-based sample, set different concentrations, set up a standard curve and get the OD? Should I do that? Who says yes? Who says no? Come on, fraidy cats. Who says yes? Should I run my beverage like I'm running a standard curve? Has to think about it. Who says no? What's the standard I'm going to run in that first assay? And all through module 1 to 5. What's the standard? BSA. That's the one you run at known concentrations. Do you know the concentration of your beverage, your solution? Well, the nutrition label says this, but I'm not so sure. We really don't know. That's my standard curve. What do I do for the line of best fit? Should I go through 0? That's a debate. Should you really? Well, it depends on what the blank is, isn't it? Is the blank a true blank? These are the questions you're going to be resolving as you go through the semester. Should it go through 0? Should it only include these 4 or 5 points? That gives me a straight line, doesn't it? And in Excel, what do you know? Click on that, it'll give you an equation of a line. Y equals mx plus b. Rearrange it, what do I get? Well, if I get an OD value for my beverage somewhere in here, I draw a line across there, do that in your books, hand draw it, I run down there, my beverage might have around 6, whatever the hell that is, protein units, whatever the hell that is. That's what you do. That's a standard curve. You're going to have to do that for all sorts of things during the semester. But that's measured at a particular wavelength. You see here it's measured at 570 nanometres. What if you have a plate rigger that can't measure 570 nanometres? Is that the end of the world? No, of course not. Can I measure it at, say, 600? Yes, but I start to have issues, don't I? What if I dilute my stain on my sample, my red dye? The more I dilute it, the less intense the absorbance. Isn't that right? Kind of logic once you get the science right. If I increase the concentration, I should increase that absorbance value. So the more there is for this red dye to stick to, the more likely I'm likely to see a sample value. Isn't that right? Now think about that, because you're going to run this spectrum down through module 3. What does my red dye do? What does my beverage do, my manufactured one? Do the two overlap so I can't measure at 570 anymore? I've got to go somewhere else. Otherwise, I'm measuring my Gatorade green or red, whatever it is. Does that make sense? That way you can modify assays. That's what your employers want. Can you see this process? Can you problem solve and adapt it to something else? While you have a will assay as a model and you're developing your own assay. That's what you do in metabolism. Problem solve this new methodology. Comfortable with that? So yes? Okay. Deep breath. Can I move to the next one? which is, as I said earlier on that urea, all these frequently asked questions, troubleshooting, the things can screw up assays. Look at them carefully, not now. Look at each one. You'll find they start to look awfully familiar. The same issues over and over. It doesn't matter what assay you're dealing with. And as I point out in the document I'll give you, even if you're doing PCRs, gel assays, the same kind of issues relate in terms of what you're doing. Can I move to the next one? So yes. All right, let's talk about the assessments in a bit of detail. The intra-session exam, I said, was take home, multiple choice, 20 of them. Usually 3 or 4 responses, answers in front of you. Once you log in, you have 2 hours. Wherever you log in is up to you. It'll be open from 9 a.m. Tuesday the 10th of September, 24 hours until 5 p.m. Friday the 13th. Now, if you think I'm going to save it until the Friday at the last couple of hours to log in and do it, that's fine. But if you get sick in the last couple of hours, you better have a medical certificate that covers you for all 4 days. Otherwise, NMP. You only get 20 questions. They'll be based on the lecture material. Well, when was that? Let's go all the way back. Be a pain in the arse. Oops, there it is. Mid-semester exam, week 8. Everything up until week 7 is covered in that mid-semester exam. Is that kind of clear? I have nearly 600 questions in catalogue that will be drawn out randomly for you. Oops. One question you will get in there is a will assay calculation. You'll be given a patient number. I will tell you the will assay, 1 o'clock Thursday prior to the mid-semester exam. This is the one we're going to use. Here's your patient number. Here's the values for the standards, the OD values. Here's the values for the wells that that patient has. You need to do the calculations. So 20 multiple choice sounds straightforward, but the will calculation, that might be half an hour or so. No going back. Go forward. Next question, go forward. All of my online stuff is no going back changing. Everyone clear on that? Open book, collude with whoever you want. Set up whatever you want. Forums, totally up to you. I encourage you to do that because these questions will challenge you to understand the work we've covered. Some will be less challenging. So you might want to make sure you're with a brains trust if you're worried. But it's testing whether you've got your handle on what's going on because not long before the final exam, eh? Do you understand the concepts we're going through? What else? I mentioned this will assay. Everything you're doing is related to that. I'm going to go through these in the lectures as well. Can I get to the next one? Right. You're going to have to write a report. Now, it's not just one. It's one, feedback, write another one. Now, this report is based on, first of all, metabolic disorders. Think of the sequence. I won't flick back again just to make you more upset. Think about the sequence. Didn't I say the first half of the semester was basically inside the cell? What genes, think about that complex pathway, what enzymes are involved in disruption to a particular disorder? Well, I have a whole list of them, of these metabolic disorders. You will come to the lab. In the lab, there'll be a whole list and you'll sign up to one metabolic disorder so that each person has their own unique metabolic disorder. Your job is to do some research on that metabolic disorder. Not in the lab, but online. You're going to look at writing up what's currently the research outcome for that disorder, what's related to it. And I mean current, last 3, 5, maybe 7 years. Keep note of that. They are headings. You need to write what's the current research of that disorder against that heading. There's the heading, research. That's what's going on in research. You're also going to look at the diagnosis, how they diagnose that disease or disorder. That's the diagnostic sector. How diagnostic people work out how to diagnose. That's a protein assay, for example. Cortisol assay is a diagnostic. Pregnancy test kit is a diagnostic. That's a threshold analysis. You're also going to have a heading called treatment. How do they treat the disease? What are the options? Not just changing their diet. What drugs do they use? How does the drug affect them? Could it be gene therapy? Some are. Altering enzyme functionality, for example. The other heading you're going to deal with is policy. What's the federal government policy? Now these, I need you to note, are the 4 key sectors that you get employment in. Basic research, applied research, translational research. The diagnostic sector, NMR, PCR, whatever it is, that's where jobs are. The treatment sector, developing new drugs, applying them. The thing about the treatment I need you to note now, and I'll come back to it later, is that you need to talk about what happens to the patient. What's their lifespan? You need to have some data to show this treatment improves or enhances their lifespan, their quality of life. And the policy sector, what's that? That's the federal government, the TGA. They say, because of this assay, we're going to treat patients. Because of colon cancer detection, we're going to have patients over 50 send in a stool sample to work out if they have colon cancer, because it's cheaper to do that and save healthcare costs than not test patients. The same with things like breast cancer. Why, when you came into the world in this country, they stuck a needle in your heel, did a blood sample to test what diseases you had initially, so they could treat you immediately, because the healthcare costs beyond that are so significant and often life-threatening. So those things are critical. But that's where you get jobs. I have many of my students, postgrads, who actually work in TGA. They are regulators saying what happens. They tell the feds this should happen. There's now a new PCR for breast cancer. Should that go into the treatment regime? Well, someone developed in research. That's now a diagnostic. But it's also used in treatment to see whether the treatment works. Does that make sense? So each of these sectors interplay. And that's important for you, because when you graduate, you need to keep a handle on where things are going. Because things evolve, some things get left behind. We no longer do some things because there's other ways to do them. New assays. Understanding how they work. So you're going to write a report on that, 1,000 words. We don't ping you if you go a little bit over or a little bit under. But if you write 2,000 words, well, you better be on song, hadn't you? You need references, and I'm going to say Vancouver. What's Vancouver referencing? You've all done Harvard. You've hammered to death on Harvard. I don't care about Harvard. That's garbage. Most publications are in Vancouver. What's Vancouver? A little number 1, that's my first reference. Doesn't matter what alphabetical symbol that is from that particular author. Number 2 is whoever comes second. Unlike my dear daughter in her first report in the US, she did Vancouver and alphabetical. That makes no sense. But you need Vancouver. Is that kind of clear? It's easier for us to examine. You'll submit that on the 30th of September, 8 a.m. Now, if you look at the schedule, back there was this thing called a Q&A tutorial. Instead of a lecture, we're going to be online. Just take note, online. Let me go back to make sure you are clear. Okay, are we clear? Face-to-face every week, except for week 8. Week 8, you go to views. On the little panel on the left, you'll see Q&A tutorials. I run a lot of these in Advanced Cell. You log in to the lecture time that suits you, either today, tomorrow, same time, 4 to 6, or the Hawkesbury session, which is what I think 9 till 11. Doesn't matter, you can do all 3. Blackboard Collaborate, has everybody used it? Has anybody not used it? Which is basically me talking, you typing in questions. Me showing you things with a shared screen, you typing in questions. Me paraphrasing your question and answering each question as they come up. I will highlight key things. I'll be doing that through the lecture as well. But that's your last chance to get some hints and tips. Make sure you're on target before you submit. Is that kind of clear? And I know you leave everything to the last minute, most of you, so that's a good time to do it. But you're not in the lecture theatre, but I will record each one of those. So after that time, when you type in a question via email, I'll say, Blackboard Collaborate, watch the recordings. You had your chance. Oop, there we go. Everyone clear on that? You submit it. I have a team of external examiners who'll mark that as quickly as they can. There's nearly 400 of you, so it takes a little while. Marking rubrics in views, you need to look at that, and I'll go through it as we go through that tutorial. Make sure you hit those benchmarks. That's what the examiners are looking at. They're experienced examiners. They've done this a number of times, so they know what I'm looking for. Current means current. Doesn't mean last year. It might be 3, 5, 7 years. What's going on in each sector? Not just pretty pictures, how are you writing? You know, you've always got these help things online to help you with your writing. Word helps with grammar. Sometimes you wonder if anybody uses Word grammar check. We done with this one? I'm not. Because you do assignment part A, we mark it as quickly as we can. We're going to give you feedback online, so you'll get a mark in views and some comments. And then we start part B. Part B is about metabolic manipulators, drugs, things like you find in grapefruit juice that screws up the pill so that you might end up being pregnant because you're having too much grapefruit juice. We'll talk about those kind of molecules. Scaring some people, that's the aim. Because unless you know what some compounds do to your metabolism, you don't know if it's going to alter it. And some drugs, some natural products screw up your metabolism. Not necessarily in a good way. You need to submit that by the 25th of October. You note that's at the end of semester. I'll be marking that as well. Feedback will be pretty limited because the aim will be to mark them, give you a mark as quickly as possible before the exam. You've already got feedback from Part A. If you haven't changed things, there's not much we can do in Part B, is there? We'll be giving you feedback in the lectures. I'll be telling you the things more broadly. I'll be putting it online as well that you should be addressing. Now I'm going to move on. The exam. The intra-session mid-semester exam was 20 multiple choice and a will assay. Calculations. Some lots of. All random questions coming at you from a huge bank, and more I keep adding each year. Because I'm trying to keep ahead of that Quizlet thing that grabs my questions, doesn't always tell you the right answer, but they make money and I don't. They may or may not help you, because often they're incorrect. The final exam, 60 multiple choice, no calculations, no will assay. I do not have calculations in the final invigilated exam. As much as I'd like to, I know the stress of the final exam can screw up a series of steps and bam, you get the wrong answer no matter how well you think you're on target. So I don't have calculations in exams. Multiple choice, usually 4 options, the answer's in front of you. I'll take you through that in the review lecture, how to tackle my exam. 30%, 60 multiple choice questions. How much is each question worth? I think you can do the sums, can't you? Short answer questions. I said there's 4 of them. They relate to the 4 topic areas we're covering. Not prognosis, treatment, policy, amino acids, fats, carbohydrates, how they might interrelate in integrating metabolism, bringing information together. You need to write. The exam booklet, not the one you write in separately, the exam booklet, the exam paper itself will have all the multiple choice, scan sheet, you've done enough of those, and pages at the end, 2 pages for each question. Question at the top, series of lines. You write in, draw whatever you need to. I'll come back to that in the review lecture. The topic areas in the review lecture to help you focus on what to prepare for. Bless you. Who's marking that? Well, we now have a reader that marks the scan sheets. Who's marking the short answer questions? Which means I've given you every lecture, 3 of them every week, and the recordings. I know what I've focused on. I've given you hints on the things to worry about in the exam. I've told you what to do in the review, 3 of them, and the recordings. And if I don't see what I know you should know, some people say, do I need to write a full page? Well, if you only write a paragraph, it better be good, hadn't it? Indeed. I will go through that. If you notice on the schedule, I won't go back again. The review lecture is focused particularly on identifying what's going to be in the exam. one of the more important ones. What does it say about the review lecture? Did you recall? Has an asterisk. When I put an asterisk on this kind of sheet, it means it's online. The review lecture will be Blackboard Collaborate, which means you can log into any session time, multiple sessions times. It's recorded. You can ask your questions. Why do we do that? Well, because often metabolism with this many students is one of the first exams off of the exam period. Your colleagues at Parramatta and particularly Hawkesbury might want to join the early session to give themselves more time to prepare. You don't have to travel in that last week. I'll remind you of that. Is that kind of clear? So yes. Any questions on that lot? Okay. Can I move to the next one? Cool. The quiz. You should be having some electronic device in front of you right now because we're going to do a practice one today. Quiz is worth 10%. That's 1% for each lecture. There's actually 12, so you could miss one, screw one up. The best 10 of your quiz attempts will count. They're only multiple choice and there's 2 of them. You'll get 6 minutes to do 2 of them. It's not invigilated. I'm going to run around and say, don't talk, don't talk. Talk whoever you want. Find a brains trust to help you out. But you have 6 minutes. Like always, question 1, get the answer, move to the second one. No going back. You need to do it right now. You need to log in to the quiz folder. If you look on views, metabolism, off the left-hand side, pull out the panel, you'll see quiz, click on it. You should see if it's behaved itself. Quiz, practice. It'll ask you for a password. If you can't get there, you're probably not using the UWS Wi-Fi link. You must use UWS. You have to be on campus. Problem for some people who are not on campus. Are we ready for the password? Is that a yes? Now, these quizzes might occur randomly through the lecture, may not necessarily be at the end. Are we ready for the password now? Hearing aids going flat, can't hear you. Can't help you with that. It's up to you. Sort it out. Okay. Borrow someone else's. You can use a phone as long as you get access to Wi-Fi. Can't help you. No. Talk to IT. Are we ready for the password now? 3-letter code, CAM, all caps. C for cat, A for alanine, M for Mark, all caps. That's it. Are we in? Yes, I can see some nodding heads. You have 2 questions. I don't hear any collusion and collaboration. Where is it?